Welcome to the Explainer. Today, we're taking a deep dive, a full architectural tour of one of the most successful life forms on Earth, the bacterium. We tend to just think of them as germs, but they are actually these unbelievably sophisticated little machines. So let's get this tour started. So here's our game plan. First, we're going to jump back in time to see how we even discovered these things in the first place. Then, we'll get into their basic design, their blueprint, before we tour their amazing outer defenses. After that, we go inside the factory, and finally, we'll look at the incredible strategies they use to survive and thrive. All right, to really get a handle on what we know today, we have to start at the very beginning. Think about it. There was a time when an entire universe of life was completely invisible, right under our noses. Our understanding was really built on the shoulders of giants. It all started way back in 1676 with Leeuwenhoek, who was the first human being to actually lay eyes on bacteria. Then you fast forward almost 200 years, and Louis Pasteur comes along and proves his germ theory, the radical idea that these little microbes actually cause disease. That idea inspired Joseph Lister to bring antiseptics into surgery, saving countless lives. And finally, Robert Koch gave us the playbook, a scientific method to prove that a specific microbe causes a specific disease. And this is where Koch's genius really shines through. He created this logical checklist, almost like a set of rules for a detective. To prove a microbe is the culprit, you have to find it at the scene of the crime, in every case. Then you have to isolate it, grow it on its own. After that, you have to show that this isolated bug causes the same disease in a new host. And for the final proof, you have to recover that exact same bug from the new victim. This framework, it turned microbiology into a real hard science. Okay, with that history lesson under our belts, let's get to the real blueprint. We're going to look at the fundamental design difference between a bacterial cell, that's a prokaryote, and one of our own cells, a eukaryote. Honestly, this distinction is the key to everything. And this chart really lays it all out. You can see our cells are big and complex. We've got a true nucleus, a little safe for our DNA, and all these different compartments, organelles, doing specialized jobs. Bacteria, on the other hand, are way smaller and much more streamlined. Their DNA just kind of hangs out in a region called the nucleoid, and they don't have all that fancy internal architecture. But, and this is a big but, pay close attention to the ribosome size, smaller versus larger. It seems like such a tiny detail, but as you're about to see, it's a critical weakness we've learned to exploit. Okay, time for the main event, our detailed tour of the bacterium itself. We're going to start from the outside in, exploring the amazing, complex layers that act as its armor, its engines, and its way of interacting with the world. First up, this stuff right here, peptidoglycan. You can think of it as the bacteria's own unique supermaterial. It's this strong, mesh-like fabric that gives the cell its rigid shape and, crucially, stops it from just popping under pressure. And here's the kicker. Our cells don't have it, not a bit. That makes it the perfect target for antibiotics like penicillin, which basically work by snipping this mesh apart and making the whole fortress crumble. Now, the way that peptidoglycan wall is built basically divides most bacteria into two huge groups. Gram-positives, they have a really thick, dense layer of it that soaks up a special purple dye called a gram stain. Gram-negatives have a much thinner layer, but they compensate by adding a whole other outer membrane, like a second wall, which keeps the purple dye out. And this simple color difference from a lab test, it's absolutely fundamental for figuring out what you're dealing with and how to treat it. And that extra outer membrane on the gram-negatives, well, it holds a pretty dangerous secret. It's covered in a molecule called lipopolysaccharide, or LPS for short. This molecule is also known as an endotoxin. What that means is, when these bacteria die and break apart, this LPS gets released, and it can trigger a massive, out-of-control inflammatory response in our bodies. That's what leads to things like high fever and septic shock. It's serious stuff. Bacteria also come with all sorts of cool attachments. Many have flagella, which are incredible. They're basically tiny outboard motors that spin like a propeller to let them swim around. Then you have pili and fimbriae, which are more like Velcro grappling hooks. They're essential for latching onto surfaces, including our own cells. And some bacteria can cover themselves in a slimy capsule, which acts like an invisibility cloak to help them hide from our immune system. All right, we've checked out the fortress walls. Now let's breach the perimeter and head inside to the cytoplasm. This is the factory floor, the bustling hub where all the real work of life, metabolism, growth, replication, actually gets done. Inside, it's all about efficiency. The cytoplasm is the watery gel that fills the cell.
The nucleoid is the command center, where the mean genetic blueprint is stored on one big circular chromosome. Scattered everywhere are the ribosomes, thousands of little protein-building factories. And then you have these things called plasmids. These are fascinating. They're like extra little toolkits of DNA that carry bonus genes for survival.
Now, I want you to just look at this number for a second, 70S. It represents the size of a bacterial ribosome. I know, it sounds like a really obscure technical detail, but trust me, this is one of the most important numbers in the history of medicine. And here's why. Our own ribosomes, the ones in our cells, are bigger. They're 80S. This size difference is an absolutely beautiful vulnerability. It lets us design drugs, antibiotics, that can specifically target and jam up the bacterial 70S ribosomes, shutting down their ability to make proteins. The bacteria die, but our 80S ribosomes are left completely untouched. It's the perfect example of what we call selective toxicity. Remember those plasmids we just mentioned? These are a huge deal. They're not essential for the bacteria's day-to-day life, but they often carry the genes for special abilities, like the ability to resist an antibiotic. And here's the critical part. They can be easily copied and passed from one bacterium to another, even between different species. This is how antibiotic resistance can spread like wildfire through a population. So, we've toured the structure, we've seen the hardware. Now let's talk about the software, the brilliant strategies bacteria use to survive in the absolute harshest conditions and to multiply at truly mind-boggling speeds. So, meet the endospore. This is one of nature's ultimate survival pods. When things get really bad, no food, too hot, whatever, some bacteria can package their essential DNA into this incredibly tough, dehydrated fortress. It's basically a state of suspended animation. An endospore can survive being boiled, zapped with radiation, and soaked in disinfectant. It can wait, dormant, for hundreds of years for conditions to get better again. And when conditions are good, bacteria multiply with incredible efficiency using a process called binary fission. It is brutally simple. The cell gets a bit bigger, makes a copy of its DNA, and then just splits down the middle. That's it. One cell becomes two, two become four, four become eight. You get the idea. It's the power of exponential growth. But here's the thing. Not all bacteria are speed demons. There's a huge range in what's called generation time, the time it takes for a population to double. A common bug like E. coli can double every 20 minutes under ideal conditions. That's why some food poisoning can hit you so fast. But at the other end of the spectrum, the bacterium that causes leprosy can take up to 20 days for a single division. Which brings up a really important question, right? If something like E. coli can double every 20 minutes, why isn't the entire planet just a giant ball of bacteria by now? Well, the answer is that this kind of explosive growth can't go on forever. In any closed system, like a lab culture or maybe even an infection, bacterial populations follow a very predictable life cycle. First, there's a lag phase, where they're just getting used to the new neighborhood. Then comes the log phase, that's the explosive, exponential growth. But eventually, they start running out of food and polluting their environment with waste, which leads to the stationary phase, where the birth rate equals the death rate. And finally, as the environment becomes just too toxic, the population crashes in the death phase. So, when you really boil it all down, the genius of the bacterium isn't about being complicated. It's the exact opposite. Their relative simplicity is their greatest strength. It's what allows them to replicate so quickly and adapt to new challenges with incredible speed. And that leaves us with a final thought to chew on. Bacteria have been on this planet for billions of years, and they've thrived by being simple, adaptable, and, through things like plasmids, collaborative. So it makes you wonder, in our own increasingly complex world, what can their strategies for survival and adaptation teach us about resilience?